Hello. Good day to you all, ladies and gentlemen. I'm Akash Maskar. I'm the principal analyst at BIS Research, and I am delighted to assume the role of moderator for this esteemed webinar. From the entire BIS Research team, I welcome you all to this webinar on precision oncology biomarkers, state-of-art technologies, and emerging strategies. When we talk about biomarkers in the fight against cancer, precision oncology has emerged as a transformative approach that aims to personalize diagnosis, treatment, and management based on the unique genetic and molecular characteristics of individual tumors. And central to this approach are biomarkers, which serve as an essential tool for identifying and predicting patients' response for specific therapies. So in this webinar, we will delve into the fascinating world of precision oncology biomarkers, exploring the state-of-art technologies and enabling the discovery, validation, and clinical implementation. Join us as we delve into this precision oncology biomarkers with our distinguished guest panels of experts comprising firstly Dr. Michael Rossi. Currently, he is serving as a Vice President of Translational Sciences or in Multidimensional Model Real-World Evidence Solutions at Concert AI. Prior to Concert AI, Mike was a division head of molecular oncology and solid tumors at SEMA4. So Mike is also a fellow of American College of Medical Genetics and Genomics, and also has held associate professor as a position of an associate professor in the Department of Genetics and Genomic Sciences at Icahn School of Medicine and at Mount Sinai in New York. So welcome, Michael. Our second guest of honor and expert is Dr. Shivi Chakravarti Kannan. He is a physician scientist with medical degree from India and a PhD in biochemistry and molecular biology from University of North Dakota. He is the founder of and CEO of Oncophenomics, which is an AI-driven preclinical drug discovery and development company. He has been actively involved in investigating tumor immunology and tumor microenvironment and developing novel targeted immunotherapies. Understanding tumor biology and computational biology perspective is something we looked at to understand from Dr. Kannan today. Welcome you both. Our third speaker of the day is our very own Mr. Nitish Singh. Nitish is a principal consultant at BIS Research. Nitish has over a decade of experience in market research and consulting within precision medicine and medtech space. I welcome you all three gentlemen to this webinar, and I hope that we get to learn a lot from you. Before we also proceed with this webinar, I request all the audience, we will have a Q&A session at the end of this webinar. I request all audience to post their questions within the chat window of this webinar. At the end of the webinar, I will relay these questions to the respective respondents whom they are dedicated to. So before, again, for the sake of, this is because for the sake, so that we can maintain a streamlined flow of events. So as we proceed, I welcome Dr. Michael Rossi to move ahead and present his findings and his work in the space.
Hi, thank you. So I'm going to talk a little bit about multimodal approaches to biomarker discovery. Multimodal is a term that's been used recently really to describe the complexity of genomics, proteomics, transcriptomics in terms of trying to understand translational science and precision medicine. Can I have the next slide, please? Yeah, and my disclosure is, I am an employee of Concert AI, and we are a health data company focused exclusively on oncology. I was formerly an employee of SEMA4, which was a spinoff from Mount Sinai. It's now GeneDx, and none of the information I'm going to present here is confidential or proprietary, so I have nothing to disclose. So my outline is pretty simple, and please load up the chat with questions. So I'm going to give you just a little bit of historical context. I've been doing genomics and biomarker discovery for about 20 years. So before it was even genomics and it was still genetics. And then I want to give you a real idea and flavor for how important it is to combine multiple aspects of data. So really that multimodal real world integration of information to truly understand what's happening with patients so that we can better understand and treat their disease. And I'm going to give you an example of breast cancer. Next slide. So historically, we've been doing precision targeted tests to understand lung cancer for almost 20 years. So it's about 18 years-ish, maybe 19 years. So 2004 was really kind of the first foray into using EGFR mutation status to treat with erlotinib. And back in the days, anybody who's under the age of 40 will think this is pretty archaic, but 20 years ago, we didn't have next generation sequencing really. It was just in its infancy. So we had techniques like FISH, which you can see where we could look for amplifications or translocations. And then most of the sequencing was done by capillary sequencing. And this was just a paper that came out. William Pao is now an executive at Pfizer. He was at Vanderbilt at the time. John Giuffredi still at Mass General. So it was really a combination of oncologists, pathologists, molecular pathologists, really early level molecular pathologists who started to see the value of doing some type of genetic testing to identify patients for therapy. And this was really kind of the start of how we did it. And you could see it was very kind of one at a time. You had to do one assay for one specific variant or one specific change, like an amplification or translocation. Next slide, please. And this just shows you kind of this was state of the art about 15 or so years ago. These were the number of mutations that we can look at, and you can see that we knew quite a few mutations that occurred in EGFR or KRAS or BRAF, and this was really how we did the testing. Next slide. And so what happened in 2008 was there was an economic downturn in the U.S. and around the world, really. And there was money that was put into stimulus money that was put in to help accelerate some of the testing. And we formed a consortium called the Lung Cancer Mutation Consortium. And it was a group of 16 major academic medical centers and a really amazing group of oncologists, pathologists, other types of really innovators in the field of precision medicine. And this was a paper that we published in 2018. I think this paper finally came out. And what it shows you is what we started to ask questions of was not just does the patient have the EGFR, but does the patient have other co-mutations in addition to EGFR? And what you can see in the Kaplan-Meier plots are that first Kaplan-Meier plot with the black, blue, and green traces. The black trace is actually the patients that had a targetable mutation. The green and received therapy. The green Kaplan-Meier is the patients who had a targetable mutation but didn't receive targeted therapy. And then the blue was patients that we didn't identify a targetable mutation. And if you look over to the right, you can see what we did with the patients that had EGFR or ALK mutations. We looked for P53 co-mutation status. And what you can see is that the patients who had much better survival in black had EGFR or ALK that was druggable, but did not have a P53 mutation. And this was still in the early days. Pembro was really just kind of coming on to the scene. We were just starting to really investigate immunotherapies. We know now that a lot of these patients with P53 mutation, co-mutational status will do much better with immunotherapies, but it was really important showing that it was much more beneficial to look at multiple mutations rather than just kind of one at a time and individually. So the next slide. And this just shows you a disease that doesn't have particularly good biomarkers, head and neck cancer. So this was a study, all this was done while I was at Emory. This was a study that I did with our head and neck group, where we did mutational analysis, and we're able to use a combination of mutational analysis plus whole transcriptome RNA-seq really to help to differentiate subgroups in head and neck cancer. So the two main subtypes of head and neck cancer are HPV positive and HPV negative, and you could use a combination of both molecular features, mutations, as well as differential gene expression really to see differences in patients. Sorry, the next slide. And so a lot of this really required not just the understanding, kind of biological genetic understanding of what was happening in tumors, but a lot of this was computational. So it really required building bioinformatics pipelines and understanding bioinformatics pipelines. And this was actually a study that we did just to show that we could build an open source workflow using the Galaxy tools. And Jeremy Gex was one of the developers of Galaxy. He's now at Oregon Health Sciences. And we showed that we could actually build an exome transcriptome pipeline. So if you can generate the data, we showed that you could actually do a full analysis. And Jeremy actually went off to Oregon Health Sciences and helped implement some of these very same analyses there for both discovery as well as diagnostics. All right, next slide. All right, so this gets me to the clinical vignettes. And so you can see here that this was a poster that I presented while I was still at Semaphore in 2021, really looking at a combination of both a targeted approach and an off-the-shelf panel like Oncomine, Thermo Fisher's Oncomine panel, and comparing that to clinical, sorry, there's a typo there. It should be exome, WES, WTS, so exome transcriptome. So when I left Emory and I moved to Semaphore, which was a spinoff of Mount Sinai, we developed a fully clinically validated New York State approved clinical test for both tumor normal exome as well as tumor whole transcriptome sequencing, really to try to understand and exploit the complexity of cancer and identify potentially new biomarkers. And one of the great things about what we were trying to do at Semaphore was really integrate data, both from the clinical side. So because we had this affiliation and it spun off from Mount Sinai, we had direct access to a major academic medical center that ran phase 1 clinical trials. And we could actually see kind of how patients were treated and layer that in with the molecular. And I just want to give you a little flavor of why that is so important, because if you look at TCGA, or you look at the Genie project, or you look at any kind of large-scale genomic sequencing project, sequencing is a snapshot in time. So there's this longevity of the patient's journey from initial diagnosis, actually from symptoms to diagnosis, to either surgery or biopsy, to treatment. And a lot of times that treatment will take kind of multiple courses where patients will be on multiple lines of therapy. And it really depends on where that sample was taken and what kind of testing was done, really to try to understand the tumor. And so if you can go to the next slide. And so one of the main take-home summaries of this study was that we were looking specifically in breast cancer, and in the 307 patients that we tested, more than 90% of patients had some clinically significant variant, and of those clinically significant variants, 41% of those had some clearly actionable biomarker. So 40% of patients that were tested had some biomarker that patient was eligible for some type of drug. The problem was that only about 13% of patients actually got on the therapy. And this is a major problem, and this really addresses why it's so important to have clinical history and clinical information. So this data wasn't really standalone. It wasn't unique. NCI Match, the paper that was published in 2020, which did a full summary of the NCI Match, which was a basket trial for all-comer tumors using the Oncomine test. They found that in breast, about less than 18% of patients actually got on therapy, and that was a clinical trial. So our data was real-world data. This was data that was actually in the clinic and what was happening. And so this is really important, and this can't be stressed enough when you're talking about precision medicine or precision biomarkers. Everybody gets very excited about the technology and what the technology can do, but it's really important that that technology meets clinical application and gets put to its best use. So the next slide. So I really just want to show you this vignette of two different patients, and what you can see here is pair 1 and pair 5. These were two patients that had more than one biopsy. So the main standard of care treatment with patients with breast cancer is using a drug called fulvestrant. It targets the estrogen receptor, so it's ER-positive breast cancer. And what we know about fulvestrant is that patients will develop resistance mutations to fulvestrant. And what I want to show you is we have this kind of idea that physicians are using this data to really drive their treatment decision-making. So can you go to the next slide, please? And I'll show you a highlight of pair 2. And so this actually shows you detail of pair 2. And what you can see is we had built software tools really to try to understand what we call the patient journey. So you can see in the graphic with the black box, there are patient visits at the very top, which are a succession of dots that all kind of cover over each other because the patient had some type of interaction with the healthcare system over many days. And then you can see the dots below that are all broken down into therapy or pathology visits or surgeries or treatment. And what I'm going to highlight is where you see the green arrow. You can actually see above that is where pathology was done. So you can see there were 3 different biopsies that were received for this patient over that time frame. And the years are down at the bottom. So you can see 3 biopsies were done and 2 molecular tests were done. And if you look, you can see there was a huge gap between when the biopsy was initially taken and the patient was put on fulvestrant. So that patient was actually put on fulvestrant before any kind of testing was done. But they knew the ER status, so that's why the patient was put on fulvestrant. But they didn't actually do the first sequencing until almost 2 years later. And then the patient had progression. The patient had actually been taken off fulvestrant and put on another therapy. And the sequencing was done after that happened. So after the biopsy, after that had changed. And so what this shows is that the physician, who is an exceptional breast oncologist, wasn't really using the data to determine therapy. The physician was really using the data to actually confirm what she thought was going on and potentially looked for other options. So can you go to the next example? And I just wanted to show you that this isn't really a fluke. And that this can happen similarly. So what you can see here is this patient. There are 2 biopsies. There's less of a lag, less of a time frame, but you can see that the line of therapy. You can see that long line of fulvestrant therapy above. That patient was actually kind of taken off fulvestrant, and the biopsy was done after the patient was taken off fulvestrant. And it wasn't really being used to determine any kind of treatment. So can we go to the next slide? And so before I get to conclusions, I always want to thank everybody and the people that require building these systems and maintaining the systems and functioning in production is way too hard, way too many people to thank. But a lot of the data and work was done by Rong Chen and Scott Newman and Mark Fink, who were my colleagues at Semaphore. And so can you go to the next slide? And I'm just going to summarize kind of what I think are the take-home points of what I was trying to show. So it's really this concept of multimodal data. So it really shouldn't be one data point. It shouldn't be EGFR alone without really understanding the context of current or prior therapies. It's really combining multiple data points to understand a complex disease like cancer. There really has to be emphasis placed on this integration of clinical information. It's pathology reports, imaging, having a lot of digital data and digital information, turning EMR and clinic notes into things that are digitable and machine readable, really to try to understand why patients are responding. What I showed you about the physician not really using the information to make treatment decisions, I really think that that's because this data isn't as accessible as it needs to be. I think future biomarkers will really be derived from complex data sets, so it's not going to be an ESR1 resistance mutation alone. It's really that context, and that's really what I was trying to show you, was the physician was integrating information and making decisions based off of other parameters rather than just the molecular. But hopefully we can start to integrate the molecular better to build better biomarkers and actually use them predictively. And this gets to artificial intelligence, which I think everybody is aware of. Hopefully, as we structure this data better and make this data more accessible to people, AI will really become the physician's assistant tool. So much like the desk reference that existed in medicine forever as a book and then moved online, it's really being able to structure information in ways that physicians can use that information, and then pharmaceutical companies and biotech can actually use that information to build the next generation of drugs. And with that, I want to thank everybody for their time.
Thank you, Dr. Rossi, for informing us about the importance of multimodal data and you sharing your findings on the research. I now request Dr. Kannan to join in and present his thoughts on this subject.
Yeah, can I share my screen?
Yeah, I hope the screen is visible, Dr. Kannan. We have already presented your slide.
It's better if I do it from the PowerPoint.
Sure.
Yeah, is my screen visible now?
Yes.
Okay, yeah. So thank you for the opportunity. So today we're talking about the precision oncology and the emerging biomarkers. So the topic I have chosen, a very favorite topic of mine, is tumor agnostic biomarkers. So this slide should have come first. Okay. So what is precision medicine? So we'll talk about personalized medicine. So what is precision medicine? So we have to sequence the DNA, RNA, and look at the protein levels. So there has to be specific molecular diagnostics are involved. Then the precision comes because of the molecular diagnosis. People kind of interchangeably use the word personalized medicine and the precision medicine, but it's not the same. Personalized medicine is a very broad umbrella term. So precision medicine in oncology, moment somebody says that, we are actually talking about some kind of molecular study, understanding the cancer at a molecular level. And the whole idea is instead of doing a one-size-fits-all approach, we want to identify what is really going wrong in this particular patient. So it's a patient-centered approach, and each individual patient has a unique phenotype and a genotype. And even within the same patient, each individual cancer, let's say the patient has 2 or 3 locations, multiple lesions, each lesion has its own unique genetic makeup. And even within the same tumor sites, there are multiple clones with different genetic makeup. So that's the whole concept of this tumor heterogeneity. So it's not like all the cells look alike, even though they're clonally proliferating. So it's not a single clone. So the mother cells, which we call as a cancer stem cells, we don't have the exact data yet, but a lot of people have done research on cancer stem cells. So the general consensus is at least there are 5 to 6 different colonies of this different clones within the tumor. So the only way to study this with new technologies like single cell genomics, where you can address the heterogeneity, but unfortunately in the clinical domain, it's not that easy because this tests are very expensive and it's not yet readily available in the clinical. So in clinical side, what we are doing is we either take a tissue biopsy or a liquid biopsy, either tissue samples like a paraffin blocks, FFP blocks, or the blood samples. And then the liquid biopsies have become very popular in recent days. Not many people know this. There are multiple types of liquid biopsies. The most common is the ctDNA, the circulating tumor DNA. And then you have the circulating tumor origin RNA, which is not, the RNA doesn't come alone. It's usually engulfed in vesicles, multiple vesicles, multilamellar vesicles called exosomes. Typically, RNA comes from the cytoplasmic origin. And the DNA comes only when the cell undergoes death, necrosis or apoptosis or some kind of cell death mechanism. The circulating tumor cells is one of the least studied and ignored by the clinicians as of now, but plenty of research R&D happening in the circulating tumor cells. That's one of our main focus areas in our lab also, but it's not yet widely accepted in the clinic. But these are actual real live cancer cells that are running around in the blood. So that is the most important, because these live cells may eventually take a prostate cancer or breast cancer. It may end up in the liver or lungs, or in case of breast cancer, it can even go to bone marrow or brain. And these circulating tumor cells eventually become the secondary cancers, the metastasis that we see later on in the stage. If we can find it at the early stage, then if we can intervene and we can identify the genetic profile of these CTCs, we can intervene and we can prevent the secondary cancers coming in the first place. So that's why it's called the liquid biopsy is called the holy grail of cancer treatment, diagnostics and treatment, because there's a lot of opportunity here. The 4 major approaches for this kind of precision oncology and biomarkers. The main, of course, the most common is the targeted therapies, where you look at a mutation like KRAS G12C mutation, and then you see there's a new drug approved now. Adagrasib is approved recently. So you match the mutation, the genotype with the phenotype and with the pharmacological drug. And most drugs nowadays, oncology, most of the pharma companies, they don't do this chemotherapy kind of one-size-fits-all drugs, because there's a lot of value in making this kind of targeted therapies. And every drug they launch in the market, it's a $1 billion market. So that's what is, even though it's only addresses a minority segment of the population, but the commercial incentives are very high. So the pharma companies have been launching left and right. Almost every day you'll see some new approval, a new drug came, or the same old drug having a new indication, some supplementary approvals, and the field has been expanding. It's like some people say it's a bubble, but it's on a very expansive logarithmic rate of growth right now. We have more than 300 different targeted therapies available now. When just 5-6 years ago, we only had only 30 drugs. Before that, we only had 3, like trastuzumab and a couple of guys. So now we have more than 300. And the next is the immunotherapies. So the monoclonal antibody space, there's a very big development happening in that space, especially the immune checkpoint inhibitors like anti-PD-1 and anti-PD-L1. So the Keytruda pembrolizumab, I'm going to cover it towards the end of this deck, is a blockbuster drug for multiple indications. Then we have this new concept of genome medicine, which is basically not just for a diagnostic purpose, but also for treatment monitoring. And one of the popular approach here is right now there is an acceptance in the clinic, which is called the MRD test, minimal residual disease test. So this is not a one-time test. You have to repeat before and after. And you are monitoring the patient, how the patient is performing throughout the course of the disease, throughout the course of the of the treatment to see if the patient is responding or not. And the surgery followed by the chemotherapy or radiation, whatever interventions we are doing, did it completely get rid of all the tumor or there's some little bit left over? So most popular in the colorectal cancer space, people have established residual ctDNA, the MRD status is very important in the management. So now instead of giving adjuvant chemo for everybody, they're recommending adjuvant chemo only for those with ctDNA positivity. And then the fourth is the hot topic everybody talks about in India right now. There are 4 or 5 companies working on it, is the CAR T cells, the cell-based therapies. But unfortunately, we don't have CAR T cells for solid tumors. It's only for the hematological malignancies like lymphoma and leukemia. But we only have autologous right now, but allogenics are coming soon. Already some allogenic CAR T cells and the clinical trials. Now beyond CAR T cells, there is NK cells, natural killer cells, and CAR-M, macrophages, and so many other. You take cells from a donor and then genetically modify that and then put it back in the patient. So instead of autologous, you go with allogenic. So that is also coming up soon. So that is a whole new webinar. I'm not going to touch into CAR T cells. So traditionally, just to give a brief background, if you're new to oncology, for a very long time, we only had 3 pillars of 3 umbrella treatments. So we had the surgery, the classics, even 2000 years ago during the Roman times also, people have been doing surgery. And then radiation therapy started after World War, once people invented radiation. And even the cytotoxic chemotherapy also came from World War. So I keep telling patients this so-called 5FU was invented by Nazi people. Hitler literally invented cancer chemotherapy, but he invented it as a biological warfare, but then it became a chemotherapy later. For a very long time, before the Human Genome Project, before the Cancer Genome Atlas, we only had these 3 options: surgery followed by chemotherapy, either in adjuvant setting, or surgery and chemotherapy at the same time, the neoadjuvant setting. There's always a debate. Oncologists are falling under 2 groups. Some people who believe in neoadjuvant, some people who don't believe in neoadjuvant. I'm in the group that believes in neoadjuvant. So I strongly believe, let's say you go to a dentist, you want to pull a teeth, right? So dentist will say, your gums are inflamed, I see infection. So why don't you take this antibiotics, come back tomorrow. I'm not going to pull your teeth today. I'm going to pull your teeth tomorrow under the cover of the antibiotics. So that is the recommended choice, because since we study circulating tumor cells, just blindly starting a patient with surgery, what happens? The cells leak into the blood. You are creating an artificial scenario of circulating tumor. The cells are being disseminated as part of the surgery, because surgery is a very invasive procedure. And certain cancers don't respond to chemo, like neuroendocrine origin, brain cancers, melanoma, head and neck cancers, cervical cancer, for example, doesn't respond to chemo. They only respond to radiation. And now radiation therapy, a lot of new things are coming up. So I'm not going to talk about that. So because of this latest advances in our understanding of cancer at a molecular level, at the DNA, RNA, and the protein level, so now we understand this concept of tumor-specific biomarkers, and now we talk about neoantigens, HER2 gene expression in breast cancer, very popular. Similarly, we talk about EGFR. So for a very long time, we only had EGFR targeted therapies, especially in NSCLC, non-squamous cell lung cancer. So 10 years ago, nobody was ordering this kind of comprehensive genomic profiles. They're only ordering this targeted sequencing for EGFR. And then later on, they added KRAS and BRAF. But for a very long time, it was only EGFR. People didn't even go for NGS. They're only doing RT-PCR. And then we had droplet digital PCR, single gene testing, single gene hand-picked mutations. Even today, for a country like India, where the insurance companies don't cover the cost of this biomarker testing, these kind of single gene or a hand-picked 8 gene mutations, 7 genes, and maybe 16 to 32 mutations, maximum of 96, because in an RT-PCR plate, you're going to put up to 96 wells. So maximum of 96 biomarkers. That's a very popular approach even today because of the cost concerns. So recently what happened, the pharma companies started implementing this targeted therapies. When they want to develop these targeted therapies, they started implementing biomarkers as one of the endpoints. So in clinical trials, we always talk about primary endpoints and secondary endpoints. How do you know if your drug works? So traditionally, we used to see, is there any symptomatic improvement? or you take a before and after a CT scan, MRI kind of things, a radiology or a pathology, different approaches. So some clinical improvement or a radiology confirmed improvement or a pathology confirmed improvement. Now the trend is using the biomarkers as a surrogate for your success or failure of the clinical trials. So that trend has started now. So that is where we see a lot of, not only in clinical space, but also in the clinical trial space, if you're working on biomarkers. There's a very big opportunity to work one-on-one with the pharma companies, because both the drug, the novel drug that they are developing, and the companion diagnostics, that's why it's called a companion diagnostics, can be developed at the same time, and both the drug and diagnostics test can go for a combined approval from FDA. So we have been focusing on liquid biopsy because we started in 2016, and that time nobody was even talking about liquid biopsy. So we were the first to start in the country, but we are not a commercially successful. We are more into R&D, and we are a very small startup self mode. Today, liquid biopsy is very popular because of big companies like Foundation Medicine, Garden Health, and now recently Grail, which is a spin-off of Illumina. They did a multi-cancer early detection test for 50 plus cancers. That's a very success story. A lot of people are talking about it. And the most important part there is it's not just the mutations. The real key, the biomarker that Grail uses is actually methylation, right? So even Freenome is also looking at that. So the next evolution of NGS is going to be methylation, CpG islands methylation. Today, nobody is doing that except for certain cases in colorectal cancer, where there's a lot of interesting stuff with the methylation stuff. Today, the routine NGS, clinical NGS, they don't look at methylation. They just look at small mutations, SNPs, indels, maybe some copy number variations. And I'm going to be talking about gene fusions and a couple of other interesting biomarkers. So here is where, if the single cell sequencing technology becomes more affordable, this is going to be a game changer. But as of today, it's only the DNA based testing is the one that's going on, and that's a huge scope for epigenomics, epigenetic kind of approaches. So in a single blood sample, so blood is like an information highway. So everything in the body has to enter the bloodstream, the live cells, the dead cells, everything, right? So you take 7.5 ml blood in an 88 tube, and these are the different things you can do with it. This is from a Nature review. I think this was from MSKCC. I think I forgot to include the link here. So here you can see you can do something called a fragmentomics. So you can look at the cell-free DNA fragments. Just by looking at the length of the fragment size, you can see the cancer patients versus the healthy patients. There is a difference in the fragment length distribution. Then you can look at the nucleosomal occupancy. For example, normal cells, the chromatin is condensed and the cells are not actively multiplying. Whereas in cancer cells, there's a lot of gene expression going on. So the lot of chromatin is now unfolded. It's exposed now. So the nucleosomal occupancy also gives a clue about, especially without even doing an RNA sequencing. Just by looking at the DNA fragments, I can tell you which genes are turned on and which genes are turned off, because we look at the TSS, the transcriptional initiation sites, that start sites. Similarly, there is TES, which is not mentioned in this diagram, the transcriptional end sites. So these are the 5' UTR and 3' UTR of the genes. If you see some methylation changes in these regions, we can tell that, okay, these genes are turned on. Maybe a P53 kind of tumor suppressor gene is turned off, but then there's a MYC or BCR-ABL kind of genes that the oncogenes are turned on in case of cancer cells. So we can understand what is the mutations or what is the epigenetic changes, the driver mutations, which are driving this particular cancer. So the nucleosomal occupancy is a very important concept. Similarly, there's something called tissue of origin. So one of the biggest challenges in liquid biopsy was people say, if I do a CT scan or MRI, I can tell you exactly the anatomical location where the tumor is, right? I can pinpoint you accurately in a three-dimensional space. But liquid biopsy is a mixed mash of everything, right? So I cannot tell you the anatomy of that. But since we have the genomic coordinates, we have this TSS and all this other regulatory information, we can use a machine learning algorithm. It's not complicated stuff. It's very simple, just a simple pipeline, bioinformatics pipeline. We can identify the tissue of origin. We can tell you which fragment came from which tissue, right? So that's a big advantage in liquid biopsy. So now gene fusions. So one of the most popular, some of my colleagues in MD Anderson have been part of this clinical trial. The most popular is larotrectinib, and then there is selpertectinib. So this NTRK gene fusions. So some of these images have been taken from the respective websites of the I'm just giving a disclaimer because I forgot to include a link here. From the respective websites, the pictures are from there. I like that branding. So always pharma companies are now becoming more and more social. They're also on Instagram and everywhere. So they have done a really nice job. So in this picture, I think something is cut out here. So in this picture, why the picture is important? You will see both adults and children playing in a beach, right? And also men and women, right? So it's because NTRK gene fusion is what we call a tumor agnostic biomarker. It's not just for adult cancers or pediatric cancer. It comes everywhere. It comes everywhere. But slowly, the clinical trials are happening and more indications are coming. So right now, we have 21 different cancers which are known to have NTRK gene fusions. Some common examples I've listed here. Colorectal cancer, the biliary, the bile duct cancers in India, we have a very high incidence of bile duct cancers, and pancreatic cancers and appendix, glioma and glioblastoma, mammary analog secretory carcinoma. That's a little bit rare. We haven't seen that in India yet. Infantile fibrosarcoma and soft tissue sarcoma. Sarcomas we have seen, but I don't know what classification because nobody tested these patients. So we have seen some cases of sarcoma. So what is gene fusion? So basically what happens is a portion of the particular gene, some parts of, well, let's say from chromosome 1 to chromosome 2, I'm just giving an arbitrary example. So here the orange, the yellow part and the blue part. So there is a partner A and partner B, right? So the partner gene can either come in the front or the back. So BCR-ABL is a classic example. The Philadelphia chromosome, and gene fusions are not new to us. Gene fusions are very commonly seen in lymphomas and leukemias. We always studied follicular lymphoma. We have the T911 translocations. It usually happens because of a balanced translocation, some exchange during it's a failure of recombination mechanisms. So when that happens, what happens is gene 1 is immediately followed by gene 2, and the promoter of gene 1 drives the expression of gene 2 also. That is the case in BCR-ABL. But in NTRK, what happens is the NTRK fusing with an unrelated gene. So NTRK is the important component. The other partner is not that important, but this fusion gives rise to a fusion protein, which is a totally different scenario than what we see in BCR-ABL. So this fusion protein becomes oncogenic. That is the whole problem here. So NTRK has three versions, the NTRK 1, 2, and 3, and all three are known to have gene fusion partners. And this is very easy to study in the NGS, but then there's a challenges with short read technologies like Illumina or IonTorrent. That's why we are working on gene fusion with long read technologies. So we are using nanopore sequencing. Actually, today is actually London calling meeting is happening right now. And right now there's another session happening there about gene fusions. So this is a very hot topic. If you want to enter into precision oncology biomarker space, gene fusion is like practically a brand new concept. It's just 2-3 drugs came into the market now. And this particular group of drugs, this group of diagnostic testing biomarkers is going to keep on growing every day. There are more than 15,000 gene fusions, for example, for EML4, ALK in lung cancer. And as people are building a database of all the known gene fusions. So it becomes a challenge from a diagnostic perspective because you don't know where the two genes are joined, where is the break point or the junction point, where it starts and where it ends, right? So if you don't know, but we definitely know where it starts because we know one side is NTRK, so you can always know NTRK is the starting point, or we know but we don't know the other gene. So either on the 5' side or 3', the forward or reverse. Becomes a challenge when you design primers and probes if you're taking an amplicon-based approach. So what happens is in US FDA, they typically approve any kind of diagnostics that has gone through the clinical trials as part of it. They call it as a clinical trial assay. So during the clinical trial, somebody like Foundation or Guardant or some company would have supported them with a custom test, which is basically a lab developed test, but it gets approved as part of the clinical trials. So the CTA, the clinical trial assay, has been designed for some known mutations already discovered earlier. So, but if the patient has a slightly different, just a little bit frame shift here and there, a slightly unknown mutation, or just a little bit upstream or downstream, a few bases upstream or downstream, this amplicon approach will not work. So this is the challenge with amplicon-based PCR-based approach, because in PCR you need to know both forward and reverse, and you also need to know the junction point. So finding gene fusions is very challenging. Also in short read technologies, the short reads are only 150 to 300 base pairs, but gene fusions can span the entire gene, which runs in kilobases, right? So minimum 5 kb, 10 kb kind of lengths. Certain genes like EML4, ALK, and NTRK, they're very long genes with multiple exons and introns. So doing it in short reads like Illumina ion torrent is very challenging, and you may end up with artifacts. That's the main challenge. So that's why we are hoping that we will have a better output with long read technologies like nanopore. In advantage in nanopore, we don't need to know both the forward and reverse. We just need to know the one partner, and we have a CRISPR-Cas9 based enrichment to capture the entire fragment. So automatically gene fusion will be pulled into the data. So Vitrakvi is a brand name for this NTRK targeted therapy. So you can see it's in lung cancer, thyroid cancer, GI cancers, CNS, sarcoma, salivary gland, pediatric cancers. So the list of indications keeps growing every day. So I like this figure from the website, so I kept it here. So you can see how the scope of this particular approach, the tumor agnostic. So tumor agnostic therapies. For every tumor agnostic therapy, you need a tumor agnostic diagnostics. And this table is for clinicians who want to know, do these new medicines really work? So one of the common questions I get from doctors is, man, these medicines are so expensive, all these new targeted therapies and immunotherapies, very expensive. Does it work? Should I really, if my patient has this gene fusion, should I even consider this medicine? So I say, look at the data, look at the clinical trial data, look at the outcomes data, right? So the disease-free survival, the overall survival, the progression-free survival, it's very promising, and we have never seen this kind of data with the traditional chemotherapy, the traditional so-called standard of care. So Vitrakvi and all these new drugs, we have never seen this kind of data so there's a significant improvement in the clinical outcomes. So comprehensive genomic profiling is what we recommend now, and everybody is now using this terminology, but I think this is a trademark by Illumina. So we don't say full name, we just put CGP test, we just abbreviate it, just like how people like there's a company called Epic Sciences, they do the circulating tumor cells. They have a slogan called no cell left behind, because in the US they have this concept of no child left behind in the educational system. Every child should get a pass. No, you should not fail the kid and ask the kid to retake the whole classes all over again. So it's called no child left behind policy. Similarly, Epic Sciences have no cell left behind because they literally look at all the cells in the blood sample. Then recently, Gordon 360, they had a marketing campaign in India. I was following them on LinkedIn. They said liquid is solid, because initially I didn't understand what is this liquid is solid campaign. I didn't understand. Then later on, I spent some more time on that talking to people. Then I understood today doctors in India and many countries, not only in India. India is a good example of all the LMICs, low and middle income countries, where the insurance companies don't cover the cost of testing. So doctors say, I still believe tissue biopsy is better than liquid biopsy. So that is a scenario in NTRK gene fusions, because one of the challenges in liquid biopsy, you cannot find gene fusions because the DNA is fragmented and the RNA is also fragmented. So it's very difficult to find gene fusions. I haven't seen anybody publish good data. People claim that they're doing it, but I haven't seen any good data coming out of liquid biopsy. So when it comes to NTRK gene fusions, at least, the preferred choice is NGS and also tissue. So tissue is preferred. They don't recommend liquid biopsy. Traditionally, people go for IHC testing. They use the pan-NTRK antibodies. So antibody-based approach. Also, there's FISH and other approaches. But these antibodies cannot distinguish between the different types of TRK A, B, and C, and they cannot tell you whether there is gene fusion, yes or no. They can only tell you the protein expression. They cannot tell you the presence of a gene fusion unless you have specific FISH probes designed for each type of NTRK gene fusions. If there are 15,000 different fusions in the publicly known, how many probes will you make? So that is a challenge. So FISH is a good approach. Maybe you can do it for the most common ones, like 50-60%, whichever the common ones, but then you only look, there's a selection bias, right? So fish, you have a selection bias. That's why we recommend go to the NGS, and all the companies are now started including this testing. And RNA testing is preferred over DNA because at the RNA level, the fusion detection is far better than detecting at the DNA level. And also now this NGS testing for gene fusions is now part of the guidelines. So this is another important point. Doctors in India and many countries, they will not just like that order a test. The very first question they ask is this test offered by a FDA approved or EMA or CEIBD, some kind of regulatory approvals? Or has this gone through clinical trials? Has there been some clinical validation? Is this part of any guidelines like ASMO, ESMO, this kind of guidelines? So now all the guidelines in every country, this list covers almost everything. All the guidelines recommend NTRK gene fusions by NGS. If the NGS is not available, let's say in African countries where NGS is not available in every nook and corner, if IHC is available, you can still go for the IHC, but IHC is only for a screening purpose. And maybe you want to do enrichment. So now these new things are coming. 10x genomics. I want to include some slides, but then I start, maybe it kind of goes out of topic. So 10x genomics, nanostring, and all these companies are coming up with this digital spatial profiling. So you can actually look at the RNA level, DNA level on the tissue biopsy without need to extracting the DNA RNA. So with this different special probes with molecular, with the fluorescent dyes and stuff. So that is coming. The digital spatial profiling is coming. So that technologies can be used. But again, those are expensive. It's maybe useful for the research purpose, not for the routine clinical right now, until the price comes down. So the next popular is the BRAF after the EGFR and KRAS. So BRAF V600E and V600K. So that 600th position in BRAF, very popular. So in this scenario, you don't need a comprehensive panel. You can just do a BRAF focused panel, a targeted panel. That is also perfectly fine. And this particular mutations, bunch of mutations, it's a hotspot. Is included as part of all the FDA approved, including the Oncomine version 3 panels. And the recent version 4 and version 5 is coming soon. Oncomine panels are now having a good representation of patient samples from Asian population. Also, several thousand samples have gone from Indian population. So if you are comparing Illumina TSO 300, 500 versus Oncomine, so my recommendation would be to go with Oncomine because it has better representation of mutations found in Asian populations. So the two drugs are very popular is the dabrafenib and trametinib. So the recent approval is for a combination. For some reason, there's a combination. I think they have different mechanism of actions. So Novartis is pushing for a combination for all these four major indications. So number one is melanoma. Then we don't have much melanoma in India, so our main focus is on NSCLC, lung cancer, and then also where the tumor agnostic comes. Any solid tumor with the BRAF V600E is also eligible for the same Tafinlar and Mekinist. So recently a doctor was planning to, we had a patient with this mutation and the doctor was planning to give Mekinist. I said, then what about dabrafenib? He said, what dabrafenib? Sir, dabrafenib is also recommended as a combination. Look at the recent clinical trial. Then he says, no, now the patient has to take two drugs, too expensive. I said, it's not like that. The reason they are recommending is the mechanism of action is slightly different between the two drugs. And when you give them as a combination, then only you see the benefit. You give them as a monotherapy, you don't see the benefit. So especially in the tumor agnostic therapies. So that's the main point here. So Novartis, again, from a pharma company's perspective, it's a double dhamaka for them. They'll make double the revenues. But then the doctors are always skeptical. If one drug can work, why should I do two drugs? Another common reason is, remember we talked about the clonal heterogeneity, how within the same patient, within the same lesion, the same tumor, you have multiple clones. When you do this kind of combination therapy, the chances of emergence of drug resistance is very low. So that's another big advantage with the combination therapies. Then the third thing which I want to talk about, just like the Vitrakvi, we have the Retevmo, the selpercatinib. Again, my close friend Dr. Vivek Subbiah was involved in all these red gene fusion studies, and this is again a very important topic. And I found this picture on their website. I like the slogan, just like I was saying, liquid is solid. So liquid biopsy is as good as solid biopsy. So that makes sense here too, Retevmo. So here they say where precision and strength meet, hit rate like never before. Okay, so this is their slogan, marketing slogan. And again, it's part of the NCCN guidelines. The NCCN recommends self-recognition. So for the number one indication is for non-small cell lung cancer and also for advanced and metastatic thyroid carcinoma, but it's not limited to these cancers. Now there are more and more indications. Red gene fusions are becoming more and more common, just like BRAF and other tumor agnostic biomarkers. Again, here the most important concept here is traditionally pathologists are accustomed to doing IHC, but the guideline clearly says immunohistochemistry is not a preferred way for treating it. So that is good news for genomics laboratories like us. So now I can go confidently talk to the pathologist, sir, no more IHC for red gene fusion. Come to me, I'll do NGS for you, right? So that's the whole idea. So why NGS, especially a comprehensive, they strongly recommend this exact same word, a broad molecular profiling. It's not, so they don't want a narrow approach, just look at RIT alone, because you have to be, because we don't know what is the other partner, right? So the second gene fusion partner could be anything else. So we know who are the partners, but some of these genes are known, some are unknown, and we don't have a good picture of what is really going on in Asian populations. So that's why we go with a de novo approach where we don't do any PCR or amplicon based testing. So you need a broad molecular profile. So there is a possibility of more than the frequency of these red gene fusions is 69% of patients with lung adenocarcinoma. So that's the majority, almost 70%. Really 3 out of 4 patients will have this. So definitely we have to test for this when there is a drug available. Why you want to waste your time giving your traditional chemotherapy stuff, and chemotherapy doesn't have a good success rate, you know. So and this figure, it shows the biomarker distribution on the right side, and also the type of testing in the Libretto trial, what kind of testing they have done. So 86% of the patients underwent NGS. A minority of the patients went for FISH, and also they have done some targeted PCR kind of approaches. So the major biomarkers in lung adenocarcinoma. So this data is from Caucasian data. So we don't have this kind of data for Asian people. This is what we're working on. So you can see KRAS. So the emerging biomarkers, the KRAS, EGFR, PIK3CA, and MEK1. And then we have the FDA approved therapies. So we have EGFR, we have KRAS G12C was recently approved. So EGFR mutations alone account for 17%. KRAS is 13%. ALK, earlier we used to do only EGFR and ALK. We used to do check for KRAS, but treatment was not available earlier, right? So we used to check for ROS1, and RET is only 2%, HER2 2%, and BRAF V600 2%. The one more biomarker recently everybody talking about is the MET exon 14 skipping. That is also very important. Now, finally, we are coming to the immunotherapies. So there's the last couple of slides. So people who are unfamiliar to immunotherapy. So there's some really nice images taken from Keytruda's website. So on the left-hand side, the very first panel, we show how a normal immune response works. So the tumor cells are expressing some biomarkers, the neoantigens, tumor-specific biomarkers, which are triggering the immune system, and the T cells are recognized that, and then they're activated. Then the CD8 positive T cells, they are the killer cells, they kill the tumor cells in the normal scenario. But in certain patients, what happens is the tumor also becomes very smart. It learns from the immune system. It says, see, the way the immune system works, it says, this is me, this is not me, the self and non-self recognition, right? So that's how it works. So now the tumor says, I'm still part of the body. You're thinking I'm a bacteria or a virus. I'm still part of the body. Do not kill me. Do not eat me. So that do not kill me signal is the PD-L1 and the signaling PD-1. So the T cells have the PD-1 receptors, and the ligand is on the tumor. I always get confused. Who has PD-1? Who has PD-L1? So PD-1 is on the T cells. PD-L1 is on the tumor. So Keytruda is an anti-PD-1. Okay, so it is an anti-PD-1 antibody, monoclonal antibody. But when you do the biomarker testing, you are not testing the T cells, you are testing the tumor cells for PD-L1. So doctors always get confused with this, right? So you're not testing for the receptor, you're testing for the ligand. So this is what happens, the tumor evasion, the tumor is telling the immune cells, even though you have found some problem with me, I am still part of the body, don't kill me. So this is called the immune checkpoint. So that is what is happening in most cancer patients. Roughly 1/3, 35 to 40% of the patients have this problem, immune checkpoints. So in that scenario, when we give a monoclonal antibody like Keytruda, which blocks the signaling, it prevents the PD-1 and PD-L1 to recognize each other, basically blocking that. So then what happens? Now the T cells are releasing the brakes, and our T cells are free to go and kill the cancer. So that's the whole. It's an immune checkpoint inhibitor. A negative of a negative becomes a positive effect. And below you can see the number of cancers. It's a very big list. So Keytruda is like a blockbuster drug, and the tumor agnostic therapies. Any cancer with the TMB high and MSI high, MSI stands microsatellite instability and tumor mutational burden. In MSI, there are two versions. Either you can directly say, look at the microsatellites, or you can look at MMR. So there's a deficiency of the mismatch repair genes. So the DNA repair mechanism, the bunch of DNA repair mechanisms. Similarly, the one slide which I want to include, I didn't have the time to include that. Just like MMR, we have the HRR, the homologous recombination repair, which is very relevant in breast cancer, ovarian, pancreas, and prostate cancer. So like just in that scenario, we call it the HRD test, homologous recombination deficiency. So in this scenario, MSI high or a DMMR. So the two different tests available in the market. Doctors always confuse what is MSI, what is MMR. They confuse. Both are the same, but you're looking at either a direct approach or an indirect approach. Now on the IHC side, one of the most today pathologists are very busy with PD-L1 IHC, but they still struggle because the guidelines are slightly different for different cancers. The two major things is the TPS and CPS. So they always get confused where to apply TPS and where to apply CPS. It's very simple. There's no formula here. It's very simple. Whichever clinical trial they used the TPS method, tumor proportion score, it's recommended there. Whichever clinical trial they used the CPS method, it's recommended. So it's nothing to do with the biology of the story. It's something to do with how the regulatory process works. And here they're given the formulas. For simple logic for lung cancer, advanced NSCLC, we go with the TPS score. And for us, for all the other cancers like metastatic head and neck squamous cell carcinoma, any gastrointestinal carcinoma, cervical cancer, also now TNBC, triple negative breast cancer, where Keytruda is being prescribed a lot nowadays, we go with the CPS score. So this is very important because there's a cutoffs for every cancer. Typically, it is greater than 1%. For certain cancers, it could be as high as 10% cutoff. So these cutoffs will get revised every year as new clinical trials keep coming. So that's why the pathology is always, it's a very subjective and a qualitative. It's not fully quantitative, it is semi-quantitative. That's why we prefer. Unfortunately, PD-L1 has to be done by IHC as per the guidelines, but we can also do PD-L1. Remember, we talked about RNA, we talked about the the tumor origin and all those things. Even in cell-free DNA, we can do PD-L1. In the RNA also, we can do it, but it is considered as a research use only test. It's not an approved test yet. So I think I already covered this. So the one important point, the MMR, the mismatch repair deficiency, is defined as at least one protein. So MSH2, MSH6, PMS2, and MLH1 has a loss of gene expression or some loss of function mutations. Either you look at the DNA level or the RNA level, or you can also do IHC also. You can also do a protein level also, but you have to clearly define either the protein doesn't exist or the protein is inactive and not working properly. Then we can conclude there is a deficiency in the mismatch repair. So to conclude, tumor agnostic therapies and tumor agnostic biomarkers, they go hand in hand. And this is just the beginning. So we have barely scratched the surface. I think the future will be literally every cancer, you will have a bunch of treatments. So even if people have not done the clinical trials, let's say today renal cell carcinoma, the question here is, should I give Keytruda? Of course, Keytruda got approved, but let's say I have a new drug, new monoclonal antibody targeting PD-1 or PD-L1. Should I give it or not? The usual response is, since the drug never tested in that particular cancer, they don't do it. It's called in indication and out of indication, right? Off-label indication. But in India and many countries where the insurance companies are not putting a lot of pressure, the doctors have the liberty to do off-label. So they can purely be driven the decision to treat can be driven by the evidence. So we recommend to practice the evidence-based medicine. If you see the tumor agnostic biomarkers like NTRK gene fusions, red gene fusions, TMB, MSI, DMMR, HRR, HRD, all these are tumor agnostic biomarkers, then you have the matching treatment. Even though that particular drug doesn't have an approval today, it's only a matter of time. Somebody will do a clinical trial, that approval is going to come. So go ahead and do it. So that is the adoption that is not yet happening, right? So when the drug is available and the patient is an eligible candidate, again, we have to follow all the ethics. So that there's ethical issues. Then the drugs are not available locally. I can't just walk into Apollo pharmacy and buy it. You have to import it from US or UK or Hong Kong or Bangladesh, right? So access to the drugs. Also the cost, for example, the sotorasib for Lumacras for the KRAS G12C costs 6 lakh rupees per month. And I have to treat the patient for a year. So that's you're talking about $100,000 or 72 lakhs, 70 to 80 lakhs treatment cost. Can the patient afford it? So that's also the cost considerations. So you always talk about more than the chemotherapy, toxicity of the chemotherapy, the financial toxicity of cancer treatment. That is a bigger problem to handle. But this is a promising area because now we are going in a better direction, even for rare cancers where studies have not happened. As long as you have the biomarker status positive, then the doctor can be encouraged to try out these new treatments. I think that's the last slide.
Thank you, Dr. Kannan, for the insightful session on tumor agnostic biomarkers and therapies. We have heard a lot about the technical insights on the biomarkers. Now I welcome Nitish Singh here to talk about the market aspects and dynamics of biomarkers.
Thank you, Akash. Thank you, Dr. Kannan. It was very insightful. So since we are running out of the time, I'll quickly drive through my slides. Talking about the clinical biomarker, as you have already, Dr. Kannan, have explained a lot about the clinical biomarkers. But on a holistic way, I would say clinical biomarkers basically refers to the molecular biomarkers, which are providing new target for, can say, disease characterization and drug discovery and development. So at BIS Research, we have done a lot of research around clinical biomarkers, their trends, key market opportunities, and what will be the overall market size of the clinical biomarkers, wherein we have considered the predictive biomarkers, the prognostic biomarkers, and the diagnostic biomarkers. I will talk around this, and this slide basically give you what is the role of the biomarkers in the personalized cancer treatment. So basically, it helps in assessing the individual's risk for developing cancer, aid in early detection, of course, determine the prognosis, basically determining the trajectory of a disease progression, and of course, help the physician in treatment guidance. So that is overall gist of the slide. Moving to the next slide, that we have encountered, we have talked to the physician, we have talked to the end users from the laboratories who are doing the biomarker tests. So from them, we came to understand that there are certain challenges in the biomarker driven test. And one of the most prominent challenge is the inability to identify the low expression level of biomarkers. Basically, when you are comparing an abnormal cancer with a normal cellular growth. This was one of the most prominent, you can say, the challenges that we have encountered. Following that, there were challenges in basically in distinguishing between a potential biomarker and a reliable biomarker that can be universally accepted. So still there are, you can say, there are challenges in identifying biomarkers, which is a potential biomarker for this particular cancer, or which is a reliable biomarker, which can be universally accepted, because there is a lot of difference in the Asia Pacific population, or you can say the North American population, or Latin, or any other. So this is how the genetic variation occurs in human population. And of course, as Dr. Kannan said, that the high cost, of course, similar as the therapy is very costly, similarly, the high cost of biomarker driven test is one of the challenge in this market. This slide, yeah, gives you a better view of the market perspective. So how the adoption of clinical biomarker-based tests is increasing throughout different geographical regions and in the different clinical areas. So if you look at this graph, basically this graph. You can see I have given a comparison of 2021 and 2022 data with respect to the 2032. So we have forecasted the market over the next 10 years down the line, and we have compared the different regions like North America, Europe, Asia Pacific, and Latin America with respect to the market size. So you can see the current market size, if I talk about in 2022, estimated to around 11.8 billion for North America. Right. And if you see the same market is going to reach around 28.1 billion in 2032. So it is, of course, a phenomenal growth, I can say. And the CAGR, the compound annual growth rate that we have estimated is around 8.73% for the clinical biomarkers market. So when I'm talking about clinical biomarkers, so that includes the different therapies, the different clinical areas like oncology, cardiac, neurology, infectious, and other clinical areas. So here I have shown the comparison as well, which is the leading therapeutic area or the clinical area in terms of clinical biomarkers. So you can clearly see that in the present scenario, oncology holds around 42% share, which is almost close to one of the highest here, followed by cardiac biomarkers. And then we have neurological biomarkers based studies and the products in the market that are being used for the detection of different diseases. So, but over the period of 10 years, over the down the line, next 10 years, the scenario will change. Of course, the oncology biomarkers will have some maturity in the market. Then the biomarkers related to cardiac and the neurological disorder and the infectious disease biomarkers, especially a lot of focus has been in the recent time after the advent of COVID, there has been a diversion of focus towards the diseases like COVID-19 and other infectious diseases, wherein scientists are uh, really exploring the new areas or the new areas for the detection of infectious diseases. And one of the is the detection of biomarkers. Right. Moving to the next slide, if you look at the technological landscape, how the clinical biomarkers are distributed with respect to the technology, as Dr. Kannan have already discussed, comparison between the NGS and ISC. So this is with respect to the different technologies that are being available for testing the different clinical biomarkers. So you can clearly see that NGS is holding the highest share, just not just because of it's a new technology, but because of the advantages that it gives, the high throughput, you can have a multiple view of the tumor heterogeneity. Multiple genes can be detected simultaneously at a time. But parallelly, if you look at the PCR, or we can say the polymerase chain reaction, that is close enough to the NGS. But over the period of time, of course, NGS is leading the market, and the technology like IHC certainly lose some share in the market down the line, 10 years down the line, if I talk about. Yeah, so this is a very interesting slide wherein I have figured out certain players or the major players in the market with respect to the areas. One is the service provider and other is the product manufacturer. So when I am talking about service provider, I am basically talking about the companies or the labs that are basically like Quest Diagnostic is one of the largest reference lab of the US. So they are providing the LDTs. So service providers are mostly here are focused on developing the laboratory developed tests. However, product manufacturers here basically focused on developing the IVDs. So you can clearly distinguish that Roche, Thermo, and Abbott. Abbott is one of the major players in terms of product manufacturers for clinical biomarkers based tests. Similarly, in the range of 5 to 7% share, the Kaizen, Perkin, Almar, and BD lies, and less than 5% share is being held by bioMérieux, Biored, and other companies. However, on the service side, if I talk about, of course, Eurofins and Roche are also one of the major service providers, and they have developed quite good number of LDTs in terms of clinical biomarkers tests that is basically identifying different cancer areas or the different disease. But if you look at the market share in terms of 5 to 7%, Myriad, Quest Diagnostic, and Agilent are fall under these category, and they are in a race to develop more and more LDTs that might be proved in the future for IVDs. Right. If I talk about NeoGenomics and Charles River, they have less than 5% share in the market. So this is all about the leading players in terms of clinical biomarkers. So this is something that I would, this slide I would like to discuss, like how important is basically the early detection of cancer. So I have compared the 2-year survival rate and the 5-year survival rate for NSCLC patient or SCLC patient, wherein the major problem, or the you can say the major challenge in the oncology space is the early detection of cancer. If you look at the data stage, if any, let's say NSCLC is detected at stage 1, the 2-year survival rate is around 97% and the 5-year survival rate is around 90%. On the contrary, if the same lung cancer is detected at stage 3, the survival rate, 2-year survival rate has been drastically fallen to 65%, and the 5-year survival rate has been drastically fallen to 41%. And similarly goes on with the stage 4. So this clearly shows that how important is the early cancer detection for the patient, for the physician, for the providers, as well as for the payers to save the money of the people. And of course, as we know that liquid biopsy is one of the area that is quite emerging and advancing at the business scenario. So we see a lot of opportunity in the space of liquid biopsy, basically for the early cancer detection. Similarly, if you look at the clinical trial data, a lot of clinical trial is going on. Around more than 4,000 clinical trials in oncology, specifically, if I talk about it, going on based on biomarker, right? Wherein breast cancer is leading, followed by lung cancer and prostate cancer. If you look at this slide basically compare the advancement, how the advancement in the cancer treatment and the cancer detection have reduced the death rate of the cancer over the period of time. So I have taken the data from 2010 to 2020, wherein you can clearly see that there is a rise in the prevalence. The rise in the prevalence is from 1.22% to 1.38% for cancer. But on the contrary, there is a decrease in the cancer death rate. So this basically shows that the society is getting aware of the cancer, the physician, and the latest technology are certainly being encountered or being implemented in the new facilities, and basically people are not hesitating to use the new technology in this space. And similarly, the regulatory bodies are giving a more and more approval for the new technologies like this. Talking about about the oncology biomarker space, if you look at the market is quite like having around 9.8 billion in 2022, which is projected to reach around 21.7 billion in 2032. This is just for the oncology space. I'm not talking about other biomarkers like cardiology, neurology, and infectious. So of course, the growth rate is still percent, but over the next year, since we have just came out with this market is being estimated after the impact of COVID-19. So there is a lot of impact after the COVID-19 on the research and development, the launch of the product, the supply chain, and other areas. So that's why the growth rate is being hampered. But I believe that this growth rate is going to be more than 10% in coming 2 years. Similarly, if you look at the distribution of oncology biomarkers with respect to different areas, then breast cancer holds around 35%, followed by lung cancer, which is 27%, and 18% for the colorectal cancer, and prostate cancer 10%. If I talk about the recent advancement in the, or you can say the recent launch by the major players. So Illumina recently launched the two-site genomic profiling test to assess tumor genes across 30 different types of solid tumors. Similarly, NeoGenomics laboratories, they have partnered with Biomarker Collaborative to ensure patients receive comprehensive biomarker testing. So yeah, that's all about this slide. And since we are running out of the time, I would like to conclude that early cancer detection is very important for the survival rate and the patient journey in a cancer. So liquid technology is like liquid biopsy, MRD testing, they are certainly helping in improving the oncology space. Thanks, everyone. I think the next part will be taken by my colleague Akash.
Thank you, Nitish, for the market-related insights. Overall, it has been a wonderful and insightful session. I'll take a quick opportunity to talk about a proud opportunity to talk about our Insight Monk platform. So, BIS presents an Insight Monk platform. This is a deep tech AI-based web platform which is designed to provide market intelligence data. Uh, so I welcome all the participants and guest speakers to do visit the platform and check out all the offerings that it provides, and feel free to access the same. Uh, Moving ahead, I think we will open this section for Q&A. Any questions, please? I request all the audience to please post them in the chat box so that we can direct them to the respondent speakers. Okay, so we have our first question. How does the integration of artificial intelligence and machine learning techniques enhance the identification and interpretation of precision oncology biomarkers? And what are the potential limitations or challenges associated with these technologies? Would anyone of the guest speakers please like to take this question ahead?
Yeah, so I think I was the one who mentioned AI in the call and the talk. So yeah, so if you are familiar with Chat GPT, I'll just give a simple example. So traditionally, what we do in bioinformatics when we build databases, you hire like thousands of people. So mostly you have companies working in Pune or Bangalore, like that, IT companies hiring thousands of people, minimum 300 people. For example, company like PD and DX or Versum, they hire hundreds of people. And every person is like a PhD or a MSc, BTech, MTech kind of a scientist. Their task is every day you go read minimum 10, 20 publications and articles and extract the information like for example, BRAF V600E, all the latest publications on this particular topic, right? So you extract that information, then you put it in a database. So this is a manual curation. That's what is happening until now. Today we have this GPT-4, the large language models. I can literally give you millions of publications, the entire PubMed repository to this kind of model. And what is usually done by 300 people for 300 days, I can do it in 3 hours. That's where the technology has gone now. So, but now we have to fine tune it because junk in and junk out, right? So we have to fine tune it to identify the focused information. So we have to bring in the human genome references and all the RefSeq IDs and NMIDs and everything. So you have to bring in this multi-omics approach so that the IDs can match, because every gene, every protein, you have that unique ID, right? So you have to match that. So that is the database. That's a job of a database guy, the database architect. So once you have that, and this cross-referencing can happen. So very easily you can pull for every mutation. You can pull all the recent publications, all the relevant clinical trials, the drugs, FDA approved or not. Everything information can be pulled out from all these diverse data sets. So this is one, we are actually working on this kind of platform. So we are calling it Variant GPT. It's a generative general purpose transformers. So it's a generative adversarial networks. It's a next iteration. We're actually using a mixed match of what Google is doing and what NVIDIA is doing and what OpenAI is doing. So these guys have made our job easier. So you don't need 100 people to do all this curation anymore. So even for the market research guys that the BAS people are doing, so you can improve the quality of the data that you get. So instead of just focusing on numbers, you can now focus on more drilling down into the insights by using ChatGPT. But if you use the one that is publicly available, that has a cutoff at September 2021. So it doesn't have any recent data. So the way we use the GPT, we build our own vector database and then we link the two. So what happens is whenever we reference something, there's a real-time training happening. So vector database is a new type of, it's a NoSQL database. Traditionally, databases are like Excel sheet formats. You have rows and columns. So here you have vectors. So you have zeros and ones, and then you have directionality of the data, which NLP terminology, that semantics, which is connected to what. Earlier it was called graph database. So vector database is a more mathematically better model than a graph database. Facebook uses graph database in their social media, how they connect it, right? They, without even you telling, they tell you, okay, this person probably studied with you in high school or college, right? So how do they find it? Because of the connections, the network. So that's a graph database. Vector database is more efficient and more powerful than the graph databases. So all the LLM guys, NLP guys are using it.
Thank you, Dr. Kanan. Audience, as we have quite moved past the allocated time slot, what we'll do is we will take up all the rest of the questions and try to respond them via an email sent to all the respondents. I thank you all again for joining us today. I thank you, our guests, Dr. Kanan and Dr. Rossi, and to have this wonderful and insightful session. I wish you all a great day and a pleasant evening. Thank you all.
Thank you, Dr. Kanan.